Hey there, health enthusiasts! In today's video, we're diving into the fascinating world of reishi mushrooms, also known as Ganoderma lucidum. These medicinal fungi have been revered in traditional Chinese medicine for centuries, and modern science is now uncovering their remarkable health benefits. From boosting immunity and fighting cancer to reducing oxidative stress and protecting against chronic diseases, reishi mushrooms are a true superfood. So grab a cup of tea, get comfortable, and let's explore the incredible potential of these mushrooms together. For centuries, traditional Chinese medicine has revered the reishi mushroom, Ganoderma lucidum, for its potent health-promoting properties. This medicinal fungus, also known as lingzhi, contains over 400 bioactive compounds, including polysaccharides, triterpenes, and peptidoglycans, which work synergistically to support overall well-being. Modern scientific research has begun to unravel the mechanisms behind reishi's therapeutic effects, shedding light on its ability to enhance immune function, combat cancer, reduce oxidative stress, and protect against various chronic diseases. The immunomodulatory effects of reishi mushrooms are well documented in both in vitro and in vivo studies. Reishi polysaccharides and triterpenes have been shown to stimulate the proliferation and activity of key immune cells, such as T and B lymphocytes, macrophages, natural killer cells, and dendritic cells. These findings suggest that reishi supplementation may help bolster the body's natural defenses against pathogens and malignancies. Furthermore, a clinical study conducted by Gao and colleagues found that Ganoderma, a standardized reishi extract, enhanced immune parameters in advanced stage cancer patients, highlighting its potential as an adjuvant therapy. In addition to its immunomodulatory properties, reishi mushrooms have demonstrated remarkable anticancer effects in preclinical studies. In vitro experiments have revealed that reishi extracts can induce cell cycle arrest and apoptosis, or programmed cell death, in various human tumor cell lines, including those of the breast, prostate, lung, colon, and leukemia. Moreover, animal studies have shown that reishi polysaccharides can inhibit tumor growth and metastasis, or spread, in mice. While more human trials are needed to confirm these findings, a pilot study by Zhao and colleagues reported that reishi spore powder improved cancer-related fatigue and quality of life in breast cancer patients undergoing endocrine therapy. The immunomodulatory properties of reishi mushrooms have been extensively studied, with numerous in vitro and in vivo studies demonstrating their ability to enhance immune function. Reishi polysaccharides and triterpenes, two of the most well-characterized active constituents, have been shown to stimulate the proliferation and activity of various immune cells, including T and B lymphocytes, macrophages, natural killer cells, and dendritic cells. In a study by Bao and colleagues, a complex glucan isolated from reishi spores was found to significantly increase the proliferation of mouse spleen lymphocytes and the production of interleukin-2, a key cytokine involved in immune regulation. Similarly, Cao and Lin reported that a water-soluble polysaccharide from reishi mycelia enhanced the cytotoxic activity of natural killer cells and increased the production of interferon gamma and tumor necrosis factor alpha in mouse splenocytes. These findings suggest that reishi polysaccharides can modulate both innate and adaptive immunity, potentially bolstering the body's defenses against pathogens and malignancies. The immunomodulatory effects of reishi mushrooms have also been observed in human studies. In a randomized, double-blind, placebo-controlled trial, Gao and colleagues investigated the effects of Ganoderma, a standardized reishi polysaccharide extract, on immune function in 34 advanced-stage cancer patients. After 12 weeks of supplementation, patients in the Ganoderma group exhibited significant increases in the percentage of CD3-positive, CD4-positive, and CD8-positive T lymphocytes, as well as enhanced natural killer cell activity compared to the placebo group. These results indicate that reishi polysaccharides can improve immune function in cancer patients, potentially enhancing their ability to fight the disease. The immunomodulatory effects of reishi mushrooms are mediated through various mechanisms. Reishi polysaccharides have been shown to bind to specific receptors on immune cells, such as toll-like receptors and dectin-1, triggering intracellular signaling cascades that lead to the activation of immune responses. Additionally, reishi triterpenes have been found to inhibit the activity of immunosuppressive cytokines such as transforming growth factor beta, thereby enhancing immune function. Furthermore, reishi mushrooms have been shown to modulate the gut microbiota, which plays a crucial role in regulating immune function. In a study by Chang and colleagues, reishi polysaccharides were found to increase the abundance of beneficial gut bacteria, such as bifidobacterium and lactobacillus, while reducing the levels of potentially harmful bacteria, such as Clostridium and Bacteroides, in mice. These changes in gut microbial composition were associated with enhanced immune function and reduced inflammation, suggesting that the immunomodulatory effects of reishi may be partly mediated through the gut immune axis. Reishi mushrooms have long been used in traditional Chinese medicine for the prevention and treatment of various cancers, and modern scientific research has begun to elucidate the mechanisms behind their anti-cancer effects. Numerous in vitro and in vivo studies have demonstrated that reishi extracts can inhibit the growth and spread of various types of cancer cells, including those of the breast, prostate, lung, colon, and leukemia. In a study by Sliver and colleagues, a water-soluble extract of reishi was found to inhibit the motility and invasiveness of highly metastatic human breast and prostate cancer cells. The extract also induced apoptosis in these cells, suggesting that reishi may help prevent the spread of cancer by promoting the programmed death of malignant cells. Similarly, Zhang and colleagues reported that a polysaccharide fraction from reishi inhibited the growth and induced apoptosis in human lung carcinoma cells, while Tang and colleagues found that a reishi triterpene extract induced cell cycle arrest and apoptosis in human colon cancer cells. The anti-cancer effects of reishi mushrooms have also been observed in animal studies. In a study by Kimura and colleagues, oral administration of a water-soluble reishi extract significantly inhibited the growth of sarcoma 180 tumors in mice, while also enhancing the activity of natural killer cells and increasing the production of interferon gamma. Similarly, Weng and colleagues reported that a polysaccharide-rich fraction from reishi inhibited the growth and metastasis of Lewis lung carcinoma in mice, partly by enhancing the activity of macrophages and increasing the production of nitric oxide and tumor necrosis factor alpha. While more human studies are needed to confirm the anti-cancer effects of reishi mushrooms, a pilot study by Zhao and colleagues provided promising results. In this study, 48 breast cancer patients undergoing endocrine therapy were randomized to receive either reishi spore powder or placebo for 4 weeks. Patients in the reishi group experienced significant improvements in cancer-related fatigue and quality of life compared to the placebo group, suggesting that reishi may help alleviate some of the side effects associated with cancer treatment. The anti-cancer effects of reishi mushrooms are mediated through various mechanisms, including the induction of apoptosis, inhibition of cell proliferation, and suppression of angiogenesis, the formation of new blood vessels that support tumor growth. Reishi polysaccharides and triterpenes have been shown to activate various signaling pathways involved in apoptosis, such as the mitochondrial pathway and the death receptor pathway. These compounds have also been found to inhibit the expression of anti-apoptotic proteins such as BCL2, while upregulating the expression of pro-apoptotic proteins such as BAX and caspase-3. In addition to inducing apoptosis, reishi extracts have been shown to inhibit the proliferation of cancer cells by modulating cell cycle regulators. For example, ganoderic acid T, a triterpene isolated from reishi, was found to induce cell cycle arrest in human cervical cancer cells by downregulating the expression of cyclin D1 and cyclin-dependent kinase 4, 2 key proteins involved in cell cycle progression. Furthermore, reishi mushrooms have been found to inhibit angiogenesis, a critical process in tumor growth and metastasis. In a study by Stanley and colleagues, a water-soluble extract of reishi inhibited the migration and tube formation of human umbilical vein endothelial cells, or HUVECs, suggesting that reishi may help prevent the formation of new blood vessels that support tumor growth. The extract also inhibited the expression of vascular endothelial growth factor, or VEGF, and basic fibroblast growth factor, or BFGF, two key angiogenic factors in HUVECs. Oxidative stress, caused by an imbalance between the production of reactive oxygen species, or ROS, and the body's ability to neutralize them, has been implicated in the development of various chronic diseases, including cancer, cardiovascular disease, and neurodegenerative disorders. Reishi mushrooms have been shown to possess potent antioxidant properties, which may contribute to their health-promoting effects. Numerous in vitro studies have demonstrated the free radical scavenging activity of reishi extracts. In a study by Xu and colleagues, a water-soluble polysaccharide from reishi was found to scavenge superoxide and hydroxyl radicals, two of the most damaging ROS, in a dose-dependent manner. Similarly, Saltarelli and colleagues reported that a reishi extract rich in polysaccharides and triterpenes exhibited strong antioxidant activity as measured by the ferric-reducing antioxidant power, or FRAP, and 2,2-diphenyl-1-picryl-hydrazil, or DPPH, radical scavenging assays. The antioxidant effects of reishi mushrooms have also been observed in animal studies. In a study by Ajith and colleagues, oral administration of a reishi extract significantly increased the activities of antioxidant enzymes, such as superoxide dismutase, or SOD, catalase, or CAT, and glutathione peroxidase, or GPX, in the liver and kidney tissues of rats with cisplatin-induced oxidative stress. The extract also reduced the levels of lipid peroxidation and protein carbonylation, two markers of oxidative damage in these tissues. Human studies have also provided evidence for the antioxidant effects of reishi mushrooms. In a randomized, double-blind, placebo-controlled trial, Wachtell Galore and colleagues investigated the effects of a reishi extract on antioxidant status in healthy adults. After 10 days of supplementation, subjects in the reishi group exhibited significant increases in plasma total antioxidant capacity and reduced glutathione levels compared to the placebo group. These findings suggest that reishi supplementation may enhance the body's antioxidant defenses, potentially protecting against oxidative stress-related diseases. The antioxidant activity of reishi mushrooms is attributed to their rich content of polysaccharides and triterpenes, which possess potent free radical scavenging and metal chelating properties. Reishi polysaccharides, particularly beta-glucans, have been shown to donate electrons to neutralize free radicals, while also enhancing the activity of antioxidant enzymes such as SOD and CAT. Triterpenes, such as ganoderic acids, have been found to inhibit the formation of ROS by chelating transition metal ions, such as iron and copper, which can catalyze the production of hydroxyl radicals through the Fenton reaction. In addition to their direct antioxidant effects, reishi mushrooms have been shown to modulate signaling pathways involved in the cellular antioxidant response. For example, reishi extracts have been found to activate the nuclear factor erythroid 2-related factor 2, or NRF2, pathway, which regulates the expression of various antioxidant and detoxification enzymes, such as heme-oxygenase 1, or HO1, and NADPH quinone oxidoreductase 1, or NQO1. By upregulating the expression of these cytoprotective enzymes, reishi mushrooms may help enhance the body's resilience against oxidative stress. Cardiovascular disease is a leading cause of death worldwide, and oxidative stress and inflammation are key contributors to its pathogenesis. Given their potent antioxidant and anti-inflammatory properties, reishi mushrooms have been investigated for their potential cardioprotective effects. Animal studies have provided evidence for the beneficial effects of reishi on cardiovascular health. In a study by Kabir and colleagues, oral administration of a water-soluble reishi extract significantly reduced blood pressure and improved lipid profiles in spontaneously hypertensive rats. The extract also inhibited the activity of angiotensin-converting enzyme, or ACE, a key player in blood pressure regulation, suggesting that reishi may help lower blood pressure by modulating the renin-angiotensin system. Similarly, Lee and colleagues reported that a water-soluble reishi extract reduced atherosclerotic lesion formation and improved lipid profiles in a polypoprotein E-deficient mice, a model of atherosclerosis. The extract also reduced the expression of adhesion molecules, such as vascular cell adhesion molecule 1, or VCAM1, and intercellular adhesion molecule 1, or ICAM1, in the aortic tissues of these mice, suggesting that reishi may help prevent the development of atherosclerosis by inhibiting the adhesion of monocytes to the vascular endothelium. While human studies on the cardiovascular benefits of reishi are limited, a few trials have provided promising results. In a randomized, double-blind, placebo-controlled study, Chu and colleagues investigated the effects of a reishi polysaccharide extract on lipid profiles and antioxidant status in hyperlipidemic adults. After 12 weeks of supplementation, subjects in the reishi group exhibited significant reductions in total cholesterol, low-density lipoprotein, or LDL, cholesterol, and triglyceride levels, as well as increases in high-density lipoprotein, or HDL, cholesterol levels compared to the placebo group. The reishi group also showed improvements in antioxidant status, as evidenced by increased activities of SOD and GPX in serum. The cardioprotective effects of reishi mushrooms are mediated through multiple mechanisms, including the modulation of lipid metabolism, reduction of oxidative stress, and inhibition of inflammation. Reishi polysaccharides have been shown to inhibit the absorption of cholesterol in the intestine while also promoting the excretion of bile acids, leading to a reduction in circulating cholesterol levels. Triterpenes, such as ganoderic acids, have been found to inhibit the activity of 3-hydroxy-3-methylglutaril, coenzyme A, or HMG-CoA reductase, the rate-limiting enzyme in cholesterol synthesis, thereby reducing endogenous cholesterol production. In addition to their lipid-lowering effects, reishi mushrooms have been shown to protect against oxidative stress-induced damage to the cardiovascular system. Reishi polysaccharides and triterpenes have been found to scavenge free radicals, such as superoxide and hydroxyl radicals, while also enhancing the activity of antioxidant enzymes, such as SOD and CAT, in the heart and blood vessels. By reducing oxidative stress, reishi mushrooms may help prevent the oxidation of LDL cholesterol, a key step in the development of atherosclerosis. Furthermore, reishi mushrooms have been shown to inhibit inflammation, another critical factor in the pathogenesis of cardiovascular disease. Reishi polysaccharides have been found to suppress the production of pro-inflammatory cytokines such as tumor necrosis factor alpha and interleukin-6, while also inhibiting the expression of adhesion molecules such as RVCAM1 and ICAM1 in endothelial cells. By attenuating inflammation and preventing the adhesion of monocytes to the vascular endothelium, reishi mushrooms may help slow the progression of atherosclerosis and reduce the risk of cardiovascular events. Diabetes mellitus is a chronic metabolic disorder characterized by hyperglycemia, which can lead to various complications such as cardiovascular disease, nephropathy, and neuropathy. Reishi mushrooms have been investigated for their potential anti-diabetic effects, given their ability to modulate glucose metabolism and improve insulin sensitivity. In a study by Sito and colleagues, a water-soluble reishi extract was found to significantly reduce fasting blood glucose and improve insulin sensitivity in obese, diabetic mice. The extract also reduced the expression of phosphenylpyruvate carboxykinase, or PEPCK, and glucose 6-phosphatase, or G.